And within Ag Canada, we have what the three of us think we're funny in calling the Prairie Weed Science Triumvirate. So a triumvirate is a political organization dominated by three individuals. We work for a political organization and there's only three weed scientists on the prairies. So we think we're funny in saying that we therefore dominate the Ag Canada Prairie Weed Science. So we also call ourselves the Harry, Ron, and Hermione of weed science. Sometimes we're maybe a little stressed when we're talking to each other. So there's myself in Lacombe at the Lacombe Research and Development Center. Dr. Sean Sharp is at the Saskatoon Research and Development Center, and then Dr. Charles Geddes is at the Lethbridge Research and Development Center. The three of us work very closely together. We talk a lot about our projects, we collaborate on our projects, we share data back and forth, which is why with a couple days notice, I can sort of steal some slides from Charles and present on his behalf. So let's jump into herbicide resistance. So in terms of our herbicide resistance surveys, the last fully completed prairie-wide resistance survey was between 2014 and 2017. So Saskatchewan was done in 2014 and 2015, Manitoba in 2016, and Alberta in 2017. That particular survey was led by Dr. Hugh Becky, formerly of Agriculture and Agri-Food Canada. Hugh left Ag Canada in, I believe it was 2017 or 2018, and moved to Australia, where he was leading the Australian Herbicide Resistance Initiative. He's actually since retired from that and come back to Canada, but he is retired. So Hugh led that last completed survey. The current resistance surveys that are ongoing are being led by Charles. So Saskatchewan was surveyed in 2019 and 2020, Manitoba in 2022, and Alberta will be done this summer. In addition to sort of the general prairie-wide resistance surveys, we also have specific kochia or post-harvest surveys. So those tend to be focused on kochia and Russian thistle. Those are sort of, this latest round was recently completed in 2021. So Manitoba in 2018, Saskatchewan in 2019, and then Alberta in 21. Looking forward, the idea is to combine the pre and the post-harvest surveys so that that prairie herbicide resistance survey will encapsulate both the pre-harvest and the post-harvest surveying. That will be done in Saskatchewan in 24 and 25, Manitoba in 26, and then back to Alberta in 27. So the data that I'm going to show you in terms of herbicide resistance status, I'm going to show you the results from the last completed survey, so the prairie-wide picture. Then I will show you the most recent results from the Saskatchewan survey. That's the only one that the screening and the analysis is completely finished. And so I'll show you those results so we can see where some of those trends are going and sort of use that for what we might expect in the other provinces as well. So this is the picture with wild oat. Group 1 resistance, again, 2014 to 2017, across the prairie provinces was at about 62%. So of the fields that had wild oat in them prior to harvest, 62% of those were Group 1 resistant. Group 2 resistance is lower at about 34%, so a third of the fields. That was an increase from previous surveys, however. Group 1 and 2 resistance is the lowest of the three sort of groups at about 27%. Now, other known wild oat resistance that I just want to mention, triolate or Avodex resistance. This particular active ingredient used to be a Group 8 herbicide. The Herbicide Resistance Action Committee, the global version of that, they've done some reclassifications in terms of herbicide modes of action. And so triolate is now considered a Group 15 herbicide. So just to be aware of that. We do have one population that had known resistance as well to Group 14 and 15 products. And then in Australia, there are confirmed populations with glyphosate resistance as well. So what were we seeing this last time in Saskatchewan? The percentage of Group 1 resistant wild oat has increased. We are up to 77% of fields. Now, this nice graph right above there shows how the percent of fields with wild oat that are resistant has increased over time. And so the square box dotted line is the Saskatchewan line, and it shows how we have almost a linear increase in Group 1 resistance from about the early 2000s. So that is increasing in frequency. It's continuing to increase in frequency over time. Group 2 resistance had a bit of an interesting trend in Saskatchewan this time around. We actually plateaued or decreased just slightly in terms of Group 2 resistance. So there was about 30% of the tested fields, so 30% of the fields that had a wild oat in it at the time of harvest, were resistant to group 2s. So a little bit of a decline or at least a plateau that we're seeing here. I'm very curious to see if that particular trend shows up in the other provinces as well and some of what might explain that. So something we are definitely watching as the screening continues in the other provinces. Group 1 and 2 resistance, again, likely related to sort of that overall group 2 resistance prevalence, also plateaued. So we're still at about a quarter of the fields that had a wild oat pre-harvest were resistant to both group 1 and group 2 herbicides. Another grassy weed species here, green foxtail. Again, going back to the 2014 to 2017 survey, Group 1 resistant green foxtail was about 25% of those fields that had green foxtail pre-harvest. Group 2 was about 11%, so definitely lower than our wild oat resistance, but still definitely present. Group 1 and 2 resistance is not as common yet in green foxtail. There was only one field in Manitoba that tested positive and 2 in Saskatchewan. So not as common to have that multiple resistance yet in green foxtail, but resistance still an issue in this species. Taking a look at yellow foxtail, this is a weed that was not typically a large problem in the prairies, and we're seeing increased prevalence of it, particularly in Manitoba. What's interesting is that for a weed that's sort of a newer problem, we're already up to about, 30, almost a third of the fields that had a yellow foxtail in it pre-harvest were group 1 resistant. Group 2 herbicide-resistant yellow foxtail was in 17% of those fields, and we already had 4 fields that were group 1 and 2 resistance. So with how quickly this resistance seems to be developing compared to, say, the green foxtail as a comparison, it seems like it might be a little bit more prone to resistance, and that resistance may actually be helping it spread a little bit. So definitely a weed to watch. Again, looking at the most recent Saskatchewan results, green foxtail, 28% of the populations were now group 1 resistant. So you see that is an increase. Again, if you look at the graph from previous surveys, that's an increase in group 1 resistance. Yellow foxtail, now we had 3 fields in Saskatchewan that were tested for yellow foxtail. Again, it was not previously found and tested in previous surveys. All 3 of those fields in Saskatchewan that had yellow foxtail tested were resistant. So again, some indication that fairly quick. Oh, did I lose you guys? Am I back? Okay.
Gotcha.
Some indication that the spread of yellow foxtail and this resistance may be somewhat linked in terms of allowing it to spread and or being quite prone to developing resistance as it spreads. So again, a weed that we're keeping an eye on here. Cleavers group 2 resistance, primarily Alberta and Saskatchewan here. Not surprisingly, Manitoba does not have as big of a population of cleavers resistance. This one was a little bit interesting in the last survey because we saw an increase in resistance in Alberta up to about 40% of fields, where it was sort of plateaued in Saskatchewan at that sort of 25% resistance. So again, something different happening there between the provinces. Really interested to see what happens in the current survey. Now, I mentioned the last round, Saskatchewan kind of plateaued. We're also looking at other cleavers resistance as well, just as a side note for one of my projects. So looking at the group 2 resistance in Saskatchewan, here is that plateau that I mentioned in the last survey, and you see quite a sharp spike in this current survey. So now Saskatchewan is up to that Alberta range of that 40-some% resistant. So this time around, we saw the increase in Saskatchewan. Will we see a similar increase again in Alberta, where Alberta is seeing more resistance than Saskatchewan or more of a plateau? Very interesting to see how those patterns play out. So again, here you can see this black solid line is Alberta, and that was essentially a linear increase where Saskatchewan had more of that plateau. So definitely something I'm keeping an eye on because a lot of the resistant cleavers in Alberta are more in my territory of central and northern Alberta. Other group 2 resistance, things like wild mustard, shepherd's purse, chickweed. It seems like every resistance survey that we do, we find additional species with group 2 resistance. So in the 2014 to 2017 survey, hemp nettle, red root pigweed, and pale smartweed were all identified for the first time with group 2 resistance. So with how easily, I guess, is probably the best way to say it. With how easily weeds can adapt or develop mutations to become resistant to group 2s, it's not surprising that we are continuing to identify more and more species with group 2 resistance. It's unfortunate and it can be a pain in the butt in terms of management, but it's really not surprising with the prevalence of group 2s in our cropping systems. Looking at group 2 resistant chickweed in Saskatchewan, so there were 10 fields, 9 of them were tested, and 44% of those fields were resistant to group 2s. Again, we have a fairly linear increase happening here, so we're seeing a fairly quick increase. Those fields where we're finding chickweed, more and more of those are being determined as group 2 resistant. Hemp nettle, again, we're up at about 60%. This is out of 5 fields. So it's a small number of fields, but again, where we're finding it, we're tending to find more and more resistance. So this is sort of the picture for all herbicide-resistant weeds in Saskatchewan in 2019 and 2020. Basically, 3/4 of the fields that were surveyed had a herbicide-resistant weed. And again, you can see that linear increase. You can see the cropland in terms of acreage that covers is quite large. And it's estimated to cost Saskatchewan farmers about $340 million annually. So there's a reason that we're surveying. There's a reason we're trying to keep on top of what is happening where, and then also trying to develop management strategies as well. Now, I mentioned that the post-harvest survey happens separately, so I want to take a look at some of the kochia herbicide resistance results. This first graphic here is the results of the 2012 survey in southern Alberta, where glyphosate-resistant kochia was sort of first identified in Alberta. Let's see how widespread it is. That was this initial survey, and I believe it was about 5% of the populations. Here's the 2017 survey, and you can see not only were we looking at glyphosate resistance, but we started having to look at dicamba resistance as well. And we did screen all of those populations for ALS resistance. And of course, I'm using the royal we here. It's really Charles and his technicians, who I know the techs are on here. So sorry, guys, I'm co-opting the we here. We've stopped screening for ALS resistance because over and over it was coming back as 100% of those populations were ALS resistant. So right now we're focusing more on the glyphosate and dicamba or group 4 resistance. So the 2021 survey, we're looking at glyphosate and group 4 resistance, the group 4 resistance being either dicamba or fluroxypyr resistance. So digging into these results a little bit, this is from the 2021 survey. So here is glyphosate resistant kochia. On the left-hand side, you can see sort of the level of resistance in the various populations that were tested. On the right-hand side, you can see how frequent it was in those counties that the populations were resistant. So the brighter red means we are closer to 100% of the populations tested from that county being resistant. So you can see there's a few that are much lower in terms of their resistance levels and some that are really dominated by glyphosate-resistant kochia. And this is what those screenings look like in the greenhouse. You can see the number of trays that Charles and his techs are screening at a time. Fluroxypyr-resistant kochia is not as prevalent yet in Alberta. It is there. You can see we've got a little bit more low-level resistance compared to some of the glyphosate perhaps. And again, not as many of the populations are as concentrated as some of the glyphosate-resistant kochia. So some of these counties are closer to say that 75% of their populations are resistant instead of essentially 100%. And we do have counties still that do not have populations that are coming back as fluroxypyr resistant. And again, a picture of the greenhouse for that screening. Now, dicamba-resistant kochia is another biotype that we are seeing. Again, some lower or more moderate levels of resistance, and again, less concentrated or less frequent in a lot of those counties. So it's occurring, but it's not as concentrated or as prevalent yet in those counties. And one more greenhouse picture here just for your viewing pleasure. So I'm going to focus here. This is obviously a slide straight from Charles. I'm going to focus on this right-hand Venn diagram here. I apologize for my dog in the background. I am working from home today due to a sick child. This is mortifying. I apologize. So what you're seeing in this Venn diagram is the percentage of populations. I apologize. Give me a moment here. I'm going to try and make my dog stop barking. My sincere apologies for that. The joys of working from home. Um, so in the Venn diagram, what you can see is that 33% of the populations are only resistant to glyphosate. Now, 5% of the populations are only resistant to fluroxypyr, 4% are only resistant to dicamba. But the total number of populations that have glyphosate resistance would be the 33% plus the 10% that are glyphosate and dicamba resistant, plus the 10% that are glyphosate, dicamba, and fluroxypyr resistant, plus the 25% that are glyphosate and fluroxypyr resistant. So we're looking at a total of about 78% of our populations that are resistant to glyphosate in Alberta. So we went from 5% in 2012 to 78% in 2021. So in 10 years, that is a huge, slightly scary, quick increase in resistance. Now, dicamba resistant, again, 4% resistant to just dicamba, 4% resistant to both dicamba and fluroxypyr, the 10% resistant to all three, and 10% glyphosate dicamba resistance. So overall, a little bit lower levels of dicamba resistance. Glyphosate fluroxypyr resistance is sort of our most common where we're seeing fluroxypyr resistant. But again, 10% of our populations are resistant to all three of those products. I'm going to focus here. This is just not my day for presentations going well with my dog and my doorbell ringing. I apologize. Give me a second, please. Oh, man. Really what I wanted to focus on here is the images of the populations on the right-hand side. And it just gives a bit of an idea of how these resistance traits differ between populations. So the graph on the left shows a bunch of different populations. The columns are dicamba fresh weight, dicamba dry weight, fluroxypyr fresh weight, fluroxypyr dry weight. And so you can see populations that, as it goes yellow, that means they're essentially dying, which is good. If they're staying blue, like the rocky V1 to fluroxypyr here, that's where those plants are still staying quite healthy. And so you see different patterns in terms of those resistances between populations. Now, another scary component here is the potential for group 14 resistant kochia. This is not yet confirmed, but you can see these images here from testing to saflufenicil, or HEAT is the trade name. And I mean, it's a fairly evident picture that makes it very highly likely that this is in fact a resistant population. Again, it's not yet confirmed. However, North Dakota is also testing suspected group 14 resistant kochia. They feel they are also very close to confirming group 14 resistance as well. So it's highly likely that we are going to be dealing with group 14 resistant kochia here in the near future as well. In terms of herbicide-resistant weeds generally in Canada, we have 8 unique herbicide-resistant weed biotypes, about 1.5 new biotypes reported each year. And you can see we've been on a relatively linear increase since about the mid-1970s. Where are these herbicide-resistant biotypes? We have 28 of them in Alberta. Manitoba is just slightly higher at 29. Ontario has the most currently at 38. So that's kind of where they are. We do have a couple scattered, on the coastal regions, but primarily in where our main cropping regions are, where our main herbicide use and main selection pressure would be. We do have a new form of glyphosate resistance in Alberta, confirmed in 2021, which is downy brome. And that is, again, work that Charles did, confirming, well, Charles and his technicians, of course, confirming glyphosate resistant downy brome in southern Alberta. And it was found in a canola crop. So another species that we're going to be keeping a very close eye on in terms of what does that mean in terms of its spread, in terms of control issues and all of those types of things. Now, why does resistance occur? It occurs due to selection pressure. So the herbicide applications that we do every year, multiple times a year, typically in our crops, put a selection pressure on to find weeds that have naturally evolved mutations to survive those herbicides. So I still get questions on, does the application of the herbicide cause the resistance? No, the herbicide does not cause the plant genetics to change. Those resistance mutations occur naturally just through the process of reproduction and outcrossing and all of those types of things. And it's just a selection pressure that those herbicides are putting on. I have been asked, can you say with confidence that we will see more herbicide resistance? And my question back is always, are we still spraying methods, and is that by far our primary method of weed control? And if we're answering yes, then yes, we are going to continue seeing more resistance because we are continuing to apply that selection pressure to our weed populations. So why does resistance make management challenging? I think it's fairly obvious, but this is just kind of a fun little demonstration that I like to do. These are the herbicide options listed in the Guide to Crop Protection, or the Blue Book, in 2019 for wild oat in each of these various crops. Now, if you have group 1 and 2 resistance, you end up crossing out all of these products. That's assuming across the subgroups, of course, but you would cross out these products as then potential options, which then leaves you with this. A very limited list. You'll notice the pre behind a lot of those products. That indicates that it's a pre-seed or pre-emergence product, which, as we know, can be a little bit more difficult to work with because it requires precipitation to get them active. Different soil characteristics can affect how they work. So it can be a little more challenging to use those products. And these are the old herbicide codes, but you'll notice group 8s and group 15s are dominant there. We have documented group 8 and group 15 resistance, group 8 being a little bit more prevalent, particularly in Alberta. So if we were to also remove those, we're really left with very few options. Now, how common is this group 1 and 2 resistance across all of those subgroups? Is this really a realistic thing to be showing you? I want to show you this particular wild oat patch that was collected in central Alberta. The cropping history and the data, this was provided by Dale Fedoruk. So you can see a heavy use of group 1 and 2 herbicides for wild oat management in this particular cropping system. Anything in red there is a group 1 or a group 2. When this wild oat patch was sent for resistance testing, this is the resistance profile that came back. Very high levels of resistance across the group 1 subgroups, high levels of resistance across the group 2 subgroups as well. So it does happen. It is out there. It is something that our producers are truly dealing with. So again, if you also then eliminate the group 8 herbicides, your list gets shrunk even more. And remember that group 8 is now actually categorized as a group 15. Is there automatically cross resistance between something like Avidex and something like Focus? That's not something we know right now. We know we have one population that had Avidex and Focus resistance. It had triolate and pyroxasulfone resistance. But aside from that one population, we don't know, is that the case? Would it be widespread? We don't know. It could be like the group 1s where you can have FOP and DIM resistance, or you can have just FOP or DIM. You don't necessarily have both. It could be a similar story with something like triolate and pyroxasulfone, but it's just not well tested or studied at this point in time. A couple nuances of resistance if you are resistance testing. When you get those results back, make sure you think about where and how you noticed the resistance and how you resistance tested or where you got that sample from. Was it a small patch in your field that you're having trouble controlling, or was it your entire field? Because when you get those results back in terms of, the percentage of seeds that you submitted were resistant to this, that, or the other thing, what does that mean in terms of the entire population that you're trying to manage? So I do want to put out there that confirmed resistance does not automatically mean that herbicide X or that entire group of products is useless. You need to think about where and how much of the field was resistant, um, what subgroups in that herbicide mode of action were resistant. Can you patch manage? What other options do you have in terms of herbicides and in terms of alternate strategies? If you're interested in herbicide resistance testing, there's a little infographic up on the right there that was put together by the Resistant Wild Oat Action Committee, and it does list the different labs and options for getting resistance testing done. And if you go to that weedscience.ca website, there are a number of other wild oat infographics if you are interested as well. So let's dive into management of herbicide resistance now. I want to start first by talking about integrated herbicide management. So if you're looking just for herbicide management strategies, using things like two modes of action, using tank mixes, using a pre and a post, so that herbicide layering strategy, those are the types of things that you want to consider implementing in your weed management system. Now, from that herbicide layering perspective, I already mentioned that pre's can be tougher to work with. But if you have group 1 and 2 resistance in crop, you really don't have a lot of other options because there's not a lot of post options in a lot of our crops. I hear a lot of complaints that some of the pre's don't truly control the wild oats. It's only suppression. It's not as good of control as what I'm used to seeing with my posts. And you're not necessarily wrong, again, depending on the moisture you get or the soil characteristics where you're farming. But suppression is much better than nothing, right? Resistance is a numbers game. And the more individuals that you're spraying with that post, the more likely you are to find resistance with that post. So having those things work together, reducing the numbers that each herbicide is being applied to is a good thing, even if it's not the level of control that we would love to see from a herbicide product. Resistance is a number games, and combining multiple tactics can be really key. So moving from that then into an integrated weed management focus. So we talked about the chemical management. We talked about, using effective modes of action and layering strategies and that kind of thing. What about these other three bubbles? What about the biological, the cultural, and the physical? I'll be very upfront. The biological is a very hard thing to do. We don't have a lot of options in terms of bioherbicides. They're difficult to develop because you're working with a living organism that you're then trying to not only mass produce, but you're trying to be able to store for a long term. You're trying to find an organism that will selectively control wild oat, as an example, and not wheat or barley or oats. Um, but ideally, it would control more than just wild oat in your field, because wild oat is not the only weed that is typically in fields on its own. So there's some challenges with biological control. That doesn't mean we're not still working on it, but there's not currently a whole lot of options for producers to really actively put that into their management strategy. So let's focus on the cultural and the physical controls then. This is one that I just want to show you. We talked a little bit about those pre-herbicides, but combining pre-herbicides and seeding rates. So this is work that was done by Eric Johnson in Saskatoon. And this is using a pre-herbicide and then looking at the impact of combining a pre-herbicide and 3 different seeding rates, 150, 300, or 450 seeds per square meter. Now, if you don't have a pre-herbicide, increasing your seeding rate reduces your wild oat biomass by quite a bit. By how much? Well, a 46% reduction going from 150 to 300 seeds per square meter and a 63% reduction going to 450. So you're getting essentially some suppression just out of increasing your seeding rate. Again, no pre-herbicide was applied in this particular study, in this particular side of the graph. If you add the pre-herbicide in, you still see that 64% reduction from increasing the seeding rate, 71% going to the high rate compared to the low rate of seeding. So again, there's still a seeding rate effect happening here. If you look at the low rate seeding rate with no pre-herbicide to where you've added the pre-herbicide, you're seeing about an 80% impact on biomass of that pre-herbicide, which makes sense for a herbicide that's registered to control wild oat. That's about what you would want to see in terms of impact on biomass. If you combine the highest seeding rate and that pre-herbicide, you're reducing wild oat biomass by 94%. These strategies work together, and they work better together than either of them do alone. So it is worth looking at combining things like your herbicide strategies and these cultural controls to really get the most out of your management strategy. Up until now, we've really been dealing with sort of an easy button of weed control. And I don't mean that in a condescending way, but herbicides have provided us a way to manage our weeds that are practical. We put them in the tank, we go out and we spray them, and the weeds die. They're economical, particularly as we've seen more and more products come off patent. They're efficient. You spray them, they die. That's pretty efficient. They're effective. Most of our products are greater than 90% control of the weeds that they're labeled on, right? So they're highly effective, and they're effective over a number of species in most of these cases. And we're very comfortable with them. They're something that we're used to using. Unfortunately, because of resistance and because of this tool not working as well as it used to, we're having to move away from this easy button weed control into a lot more complex, possibly time-consuming type of weed control strategy. So I want to talk here about silaging. A lot of these studies that I'm going to show you are focused on wild oat. It's where a lot of my work focuses. It's where a lot of my predecessors' work focused. So this is kind of what I'm going to focus on for the management side of things. So let's talk about silage, which is essentially cutting wild oat at panicle formation. So this is a study that Neil Harker conducted, looking at barley grown for grain. This is the black line with no herbicides, pretending those wild oats were resistant. What happens to the wild oat population if you're not spraying them at all? If you're producing barley for grain using a full rate of registered herbicides, you definitely reduce the wild oat densities. However, if you did barley for early cut silage, so within about a week of head emergence is how this study was set up, you could actually decrease your wild oat densities below barley for grain with a full rate of herbicides, and no herbicide was applied in this early cut silage treatment. So that can be a really effective management strategy for wild oat because you're cutting it before those viable seeds are produced. Neil then went on to look quite in depth at integrated wild oat management. So we're going to look at one of his studies here. He had direct seeding rotations that were continuous barley versus a barley canola barley pea, so a more diverse rotation. He had two varieties or cultivars, a short versus a tall, with the tall in theory being at least more competitive, using two seeding rates, 200 to 400 seeds per square meter, and then using a quarter, a half, or a 1x of herbicide rate. This is when there was a lot more interest in cutting herbicide rates. We're not going to focus on that component. For our purposes, we're going to pretend that a quarter rate was just dealing with a resistant population, so the herbicide didn't work as well. Those treatments were applied to the same plots year after year, and so we're looking at cumulative treatment effects. So if you did one of those factors, you increased your seeding rate, you used a more competitive variety, or you diversified your crop rotation, you could reduce wild oat biomass by about 2 to 3x. If you did two of those things in combination, remember I said doing those things in combination works better, you could reduce your wild oat biomass by 6 to 8x. If you did all three of those things in combination, the reduction in wild oat biomass was about 19x. So again, adding those strategies together, they work better together than any of them do on their own. Neil then wanted to do a new study with more diversity, combining optimal cultural wild oat management tactics with truly diverse rotations, not just summer annuals. So he had 14 treatments, including a 100% herbicide, a chem fallow, and an alfalfa sort of check or comparison treatments. All plots did receive a full rate of dicot herbicides. Again, in this particular study, he was focused just on wild oat. So what did he see here? This is wild oat emergence or wild oat densities. And at the bottom here, circled in red, is the canola wheat, canola wheat, canola rotation that had 100% of wild oat herbicides. So it had full herbicides all the way through. And we were at about 32 wild oats per square meter. Now, if you look at some of these treatments, this would be a 2x seeding rate of early cut barley silage, 2x seeding rate of winter wheat. We had some winter triticale, and then we had our 3 years of alfalfa treatment up here. So looking at the 3 years of alfalfa treatment, we were statistically similar to that canola wheat with 100% herbicides. However, in those alfalfa years, there was no wild oat herbicide applied. So 3 years without wild oat herbicide, and yet still a very similar density. Similar story here with the early cut silage and our winter cereals, 3 years of no herbicides, and yet we were still able to maintain those wild oat densities similar to that canola wheat rotation with full herbicide rates. So it is possible with multiple strategies to actually remove completely a wild oat herbicide and still maintain those densities. Now, wild oat don't occur in a vacuum. This is building on Neil's research, and he always loved Far Side comics, so this is sort of a shout out to him by including this Far Side comic here. But when I started with Neil, I wanted to look at what about other weeds, not just wild oat. And so we developed a 5-year study where we included a lot of those strategies that Neil had seen were effective: perennial forages, 2x seeding rates of spring barley, barley silage, winter cereals, and we also added in chaff collection. Now, chaff collection was used to simulate any harvest weed seed control strategy in this particular study. I just want to show you some of the results. This is across multiple sites. I'm not showing the 2016 and 2020 treatment because they were the same across all of our treatments. It was a 2x wheat with no herbicide and no chaff collection. This is sort of our treatment list, similar to how Neil had it set up. Our treatment's over here, our graph will be in the middle. We're going to essentially just ignore the site compliance graph for today. But what that does, if you're interested in looking at it as I'm presenting these results, it shows you how many of those sites had that response, how many had that response but were not significant, or how many were significant when maybe the graph shows no significance. If we look at wild oat density here, this yellow bar is from our wheat, canola, wheat rotation with 100% herbicide. So this is our baseline comparison treatment. These treatments that I have boxed are where we included full herbicide applications. So these had broadleaf and wild oat herbicides in each year. And you'll see all of these had very similar densities to this canola wheat rotation with 100% herbicides. So where herbicides were applied, our wild oat densities stayed very similar. These rotations here that now have boxes on, these are where we pulled out the herbicides and we were trying to just use some of our integrated weed management strategies to compensate for that. In these particular ones, you'll notice the red bars here. That's where our wild oat densities were significantly higher than that canola wheat rotation. However, I want to highlight these treatments here. So again, 3 years of alfalfa without herbicides. Treatments that included 2 years of silage barley plus a competitive winter cereal all had statistically similar wild oat densities to that canola wheat full herbicide treatment. Now, I specified a competitive winter cereal. We did have winter cereals down here. We had winter wheat. At quite a few of our locations, we had winter kill issues. And so our stands were not super competitive. And so we didn't see the same impact of the winter wheat in this specific study as we did winter triticale and fall rye, which survived our winters better across our locations. Now, I mentioned wild oat doesn't occur in a vacuum, so we looked at wild buckwheat density as well. Now, interestingly, we didn't see an impact of any of our treatments on wild buckwheat. And to be honest, this is something that I was a little bit concerned about with wild buckwheat, because if you increase your seeding rate on wild buckwheat, it's just going to climb up the plant higher to get the sunlight. I don't think it's going to respond as well to some of those increased crop competitiveness strategies as, say, a wild oat does, that doesn't have the ability to climb to capture the light and capture the space. So there's more work that we need to do on wild buckwheat to understand what integrated weed management strategies might work on it. And that goes for other twining weeds like cleavers as well. Now I want to talk very briefly about the broadleaf weed biomass. When I went to analyze this and look at the results, my low diversity, low seeding rate, no herbicide, no chaff collection treatment had reduced broadleaf weed biomass, which made absolutely no sense to me. That is not how that should work. Then I went back and looked at some pictures and I realized what had happened. This is our canola wheat rotation. This is obviously a canola plot with 100% herbicides. Looks pretty clean. What happened in some of the other rotations was that the wild oats were so competitive that they outcompeted the broadleaf. So this is in Lacombe. There's the canola with herbicides. There's the canola without herbicides. That is actually a canola plot, but you can see just how dense our wild oats were. So that's why we had a weird response with some of our broadleaf biomass. Crop biomass, again, similarly, where we pulled our herbicides and didn't go diverse enough in our weed management strategies, we did see reductions in crop biomass. We did have an increase in biomass here where we diversified but included our herbicides, so you can see a benefit of that happening there. And again, our alfalfa, our silage barleys, and our competitive winter cereals resulted in statistically similar crop biomass. Now I want to highlight this one just for one quick sec. This is a canola wheat rotation where we added chaff collection. So we kept our herbicides, but we added chaff collection. You see a non-statistically significant increase in crop biomass, and I'll show you why I'm highlighting that right now. We actually saw a yield increase in that particular treatment. So we're seeing an impact, I think, of that chaff collection where those herbicides are still being effective. There's an added benefit of that chaff collection in that particular treatment. We also saw increased yield in the alfalfa treatment as well. Again, 3 years of no herbicides there. Poor yield, unsurprisingly, you saw the wild oat densities in Lacombe, where we pulled herbicides and those integrated weed management strategies were not enough to compensate, but better in terms of our yield where we had our silage barley and our competitive winter cereals. Now I just want to show this quickly. This is looking at the wild oat seed bank. This is in Saskatoon where you can see the benefit again of some of those integrated weed management strategies on the wild oat seed bank. In Lacombe, you're seeing more negative impacts where we couldn't compensate for the loss of some of those herbicide applications. So what's the difference between the two sites? It's sheerly the density. So in that canola wheat rotation with 100% herbicide, Saskatoon has about 3,000 seeds per square meter. That same treatment, Lacombe has about 12,000 seeds per square meter. And so I do think there's a density impact here, which is suggesting to me that adopting IWM strategies before your weeds get really dense, you're going to have better efficacy of those IWM strategies. So again, just summarizing that particular study, using a 2x seeding rate of silage barley with competitive winter cereals, you could maintain your yield with 3 years of no herbicides. 3 years of alfalfa increased the yield, which was really positive to see. Chaff collection seems to increase yield when it's sort of that incremental addition to your weed management strategy. We need to look more at twining weeds, and those initial weed densities are impacting IWM success. Talk a little bit about harvest weed seed control. I had a field scale study with the Harrington Seed Destructor. I've been really frustrated trying to show the results of that study because there's generally a lack of significance. We didn't have a lot of impact on those weed densities. Now, why not? If I just showed you that chaff collection can be beneficial, why wouldn't these mills be working? Part of it was the field location. We went to the dense weed patches in the field, which is sensible. That's where our weed densities are going to be most consistent. However, it is also where it takes the longest time to have an impact on those densities, and this was a fairly short-term project. It was about a 3-year project. So I think it was not long enough for as dense as some of those weeds were. I think we might have some unaccounted for losses in terms of seed retention of our weed species. So at the crop or at the combine header, as an example, we haven't accounted for the loss of seed retention there. And so we maybe weren't getting as much efficacy as we thought we were from some of the preliminary studies. And then in Australia, they've done some work that shows combine settings can impact how much of your weed seeds are actually going into the chaff and therefore into the mill. We did not use specific combine settings in this study. My goal for this study was to be giving the farmer clean grain samples off of our study area so that the farmers would let me come back for the next 2 years of the study. And so we optimized our combine settings to try and give them clean samples over optimizing combine settings for concentrating weed seeds in the chaff, and that may have impacted some of our results as well. So I don't have definitive data showing these things working, but I am also not ready to say that they don't work either, because I think there was maybe some things that limited our ability to look at how well they worked in this particular study. Having said that, we've talked a lot about harvest weed seed control and wild oat over the last few years, and how it may not be a great option for managing wild oat densities because wild oat loses so much of its seed before the crop is mature. However, one thing that we are looking at is whether or not harvest weed seed control would allow us to keep wild oat patches as patches. Because even though there's not a lot of seeds left, those seeds that are left, we're spreading across the field. And some of the research that Steve Shirtliff did in the early 2000s showed that, he measured as far as 250 meters and was still finding wild oats being spread. And that's as far as he measured, not necessarily as far as they spread. Um, so we know those combines can spread the wild oat patches. And if you have a patch of wild oat, why is it there? Well, there's a good chance it's resistant, and or it's quite competitive, and or it's coming up at a time that is not matching up with your management strategies. Either way, it's really genetics that you don't want to spread throughout your field, because then more and more of your field is going to have wild oats that are hard to manage. So we're looking at harvest weed seed control. It's an ongoing study right now to try and see if that could at least help us keep a wild oat patch as a patch, where you could then employ patch management strategies instead of an entire field management strategy. I get some questions on row spacing and openers. There seems to be a bit of a movement from sort of a 9 to 12 inch row spacing to a 15-inch row spacing with some of the newer seeders. As a weed scientist, to me, narrower rows are better. I prefer the 9 to 12-inch spacing versus a 15-inch row spacing. Now, that is just me speaking as a weed scientist. I can't speak to, efficiencies on farm or logistics or any of those things that farmers are actually dealing with. But from a weed science perspective, I would prefer us to keep it narrower. I also get some questions on different openers, a knife opener versus a paired row versus a disc. Now, disc openers are lower disturbance. So in theory, you should see less weed emergence because you're not disturbing that soil. You're not burying some of those weed seeds and helping them emerge. Having said that, we haven't done those studies in Western Canada as of yet. So I don't have data on that, but I would suspect that a disc opener would actually result in lower weed emergence. A paired row, it's a lot harder to say, and there's likely some impact of the environment in that year. You do have more disturbance because it is a larger opener, but you're also spatially distributing the crop. So if the crop establishes really well and is really competitive, that increase in disturbance may be outweighed by that crop competition. If your crop is not establishing super well, we don't get a lot of rain, it's a weaker stand, then that increased disturbance may give the weeds the advantage. And again, it's not studied. This is just me sort of working through the theory of how these things are working in the field and what might be happening. So I don't have a great answer on the openers, particularly the paired row, but that's kind of my 2 cents on some of that. Wrapping it up, in terms of sustainable weed management, redundancy is key. You want to be targeting every weed species with multiple management strategies, because it's harder for a weed to develop a resistance to all the strategies than just one single dominant strategy. If you're using multiple strategies, chances are one of those strategies will help you manage that species or that individual. If you just have one dominant strategy, it's easy for weed population to shift and start to evade that strategy. Please don't just rely on new herbicides or modes of action. That ends us up on the herbicide treadmill with resistance. It's sort of what we've seen with, we had group 1s for wild oat, and everyone used group 1s and we started getting resistance, but the group 2s came out. So more adoption of the group 2s, but now we're just getting group 2 resistance. It's what you see a little bit with kochia. We had ALS herbicides, we started getting ALS resistance, we relied more on glyphosate. Then we got glyphosate resistance. Group 4 use went up to manage kochia. Now we're seeing group 4 resistance. As that Group 9, Group 4, Group 2 resistance spread, there was more use of the Group 14s, and now we've got suspicious populations for Group 14 resistance. There's a treadmill. If we're just relying on a single new mode of action, there's such a high selection pressure on that we just end up on this treadmill of new product, resistance, new product, resistance, new product, resistance. And we can't quite get ahead of that. We just get stuck in that loop. So using those alternative strategies, using redundancy in our weed management strategies, that there's multiple strategies hitting the same weed over and over in that cycle, is your best bet to keeping your weed management sustainable. Just to emphasize that, this is the timeline of introduction of new herbicide sites of action. So you can see we did really well for about 40 years, and then it took about 30 years for that one new group to come out. And as that one new group comes out, are any of those actives actually, being registered in your crop for your particular weed of interest? I'll end it there. My contact information is there if you want to get in touch with me, as is the Wild Oat Action Committee site if you want to take a look at any of those infographics. So if there's any questions, I'm happy to take them.
Thank you, Breanne. We do have some questions in the chat. I'll maybe just start at the top of the list. Are there suggested products for kochia?
Ooh, that would be a Charles question. I don't deal with kochia as much in central Alberta. We also don't typically make product recommendations, but he might have some suggestions on sort of herbicide groups, although I believe we're in a very similar situation as we are in wild oat, where there's not necessarily a lot of options left. So I guess what I would say is whatever might still work.
Another question, is there any new grassy weed chemicals in the pipeline at all that you know of with different grouping than what's available on the market today?
The newest one that I'm aware of that is actually coming to market would be tiafenicil, which is a new group 14 from Gowan. Aside from that, I am not aware of any new herbicide groups that should be coming to market in the next couple of years. Not to my knowledge. Not in Western Canadian crops anyways.
And then a question about seedbed utilization and any research that's ongoing.
I'm off the very top of my head, I'm not aware of any projects that are ongoing. We've talked about looking at those openers in a few different projects and still figuring out the right way to do it and the right way to incorporate all the different questions that we want answers to. So it's on minds and it's in the pipeline, but I'm not aware of any off the top of my head that are ongoing right at this second. But don't quote me on that because there's always a lot that I'm not aware of ongoing. So.
Regarding Avodex, what's the best time to apply? Fall after combining or in the spring before seeding?
I would defer a little bit to the actual chemical company on that who works more with the actives. I would say in general, with pre-applied products, when there is a fall application, you get a little bit more guarantee of some activation moisture just from the snow melt, where if you're applying in the spring, you are really crossing your fingers and hoping and praying that you're going to get those spring rains, which, as we've seen in recent years, may or may not come until after some of those weeds are emerging. So I'm not confident on that as a product specific thing, but generally with soil applied herbicides, at least you know you're going to hopefully have some snow melt activation moisture where you're not as confident on getting those spring rains.
And a tag on to that question, should you harrow it and then seed, or is seeding enough to embed it within the soil?
That's not one I can really comment on. That would be field specific, product specific. I would talk to your Gowan reps and take into account sort of what openers you're using and things like that. So
All right, so the first question is up and we have a decent amount of responses, and the majority of people are saying that more frequent for herbicide resistance in the future, obviously.
Yep, and I would agree with that, particularly as we continue to use herbicides as our dominant strategy.
Well, as funny as I think it is to give up, nobody's picking that one as the answer.
I'm really glad that no one's picking that no one's feeling quite that defeated.
Yeah, everybody obviously is using the integrated weed management.
Excellent.
Um, so seedbed utilization or seed density to control weeds.
Oh, Okay, that's an interesting question. So we have more consistent results where increased seeding rate is impacting weeds than we do from row spacing or seedbed utilization. I'm gonna say row spacing here. However, we do see research from, say, Denmark is coming to mind, where they also showed that if you do the combination of seeding rate plus sort of your seating uniformity, I guess might be the best word for their particular study, that it was actually enhanced control as well. So we tend to see seeding rate be more consistent. That's not to say that seed bed utilization or row spacing doesn't have an impact. We typically see row spacing impacts be narrow is better. In the research study, it's narrow is better or no difference, depending on the row spacings utilized. Um, and again, in terms of, knife versus um, paired row and those kinds of things, we don't really have that data yet. I see another question there. Has there been much work done with soil disturbance? Um, I'm going to go back to sort of the switch from conventional tillage to no-till. We consistently saw reduced weed densities as we moved into no-till or min-till systems. So increased soil disturbance typically stirs up that weed seed bank. You get increased seed soil contact with a lot of those. Some of those seeds that were on the surface and not able to germinate unless buried get buried and then start germinating, things like that. So disturbance typically increases weed seed germination. Um, Again, that's where sort of those paired rows are an interesting question, because you've got increased disturbance compared to, say, a knife opener, but you've also spread that crop competition out more. Do you need to do an increased seeding rate to adjust for that difference in how that crop is distributed? Is the crop competitive enough to compensate for being spread out and for that increased disturbance? Those are all questions that I don't have answers to. It's questions I'd really like to find answers to, but we haven't done those studies yet, so I don't have an answer. But generally, increased disturbance equals increased weeds. So a quick comment on this one, just based on the answers. The pre-post tank mixing layering, those are all good weed management strategies, but that would be part of an integrated herbicide management strategy only, or just chemical management of weeds. A true integrated weed management strategy would have the physical, cultural, chemical, and biological aspects.